Enter, what is that? AirPlay device password. I don't know what that is. I hope that this is still recording correctly. Okay. Okay. All right. So, welcome to lesson 6. This is part 2 of protein synthesis. And we are going to start by doing a review of the concepts that we have discussed before. Okay. So, Maria is going to lead us in that part of the discussion, and this is basically going to review some of the things that we learned last time, okay? So Maria, what are the 4 building block sets of living organisms?
The 4 basic building blocks of organic compounds were lipids, nucleic acid, amino acids, and carbohydrates.
Excellent. Yeah, and what are the carbohydrates used for?
The carbohydrates are used to store energy like sugar and starch.
Excellent. And you gave me some examples right there. Do you know any names for this?
Names for which one?
For like sugars and starches and things like that. If you remember. Okay, like for example, one very common is glucose. Okay, glucose, fructose, galactose. Galactose is the sugar that is in milk. Very interestingly enough.
Oh, okay.
Okay. Yeah. All right. So the wind sound that we have, I think that is Maria's microphone rubbing on something, but that's okay. Okay. Aubrey says cornstarch. Cornstarch is also a carbohydrate. That's right, and it's one of those starches. Cellulose. Good. Excellent. Cellulose is another carbohydrate. Okay. What are lipids used for, Maria?
They're used as building blocks of the membrane and the energy storage.
Excellent. Yeah. And can you give me also some examples of lipids? Well, you mentioned the cell membrane, okay?
Yeah.
What were the names of the lipid that is used in the cell membrane?
I can't remember.
Okay, anyone else that remembers? The name for that, it had a long name, okay? Phospholipids. Okay, remember that when we were talking about the phospholipid bilayer, okay? So I'm going to put Emmeline for number 5. Okay, so something interesting that I've learned about lipids, lipids are fats and so on, and a lot of people kind of give the lipids a bad name, I would say. I wouldn't, people say, oh, you don't want to eat too much fat and so on. But interestingly enough, the nervous system, so all the cells that compose the nervous system are very, very rich in lipids. So most of the fat in our body, most of the functional fat, the useful one, is actually in the brain, all right? So, so it is really, really important that we give our body also the proper amounts of lipids in order to build and to function correctly. So Aubrey says fat doesn't make you fat, sugar does. So fat is very healthy. So everything in moderation, isn't that right? Okay, so if you eat too much of anything, it would be bad. Okay, even water, too much water could kill you. Okay, so let's continue, Maria. What are nucleic acids used for?
They store the organism's genetic code in DNA and RNA structures.
Excellent. Excellent. And there are basically 5 different types of nucleic acids. Do you remember what are those?
Yeah, adenine, cytosine, guanine, thymine, and the last one. I can't remember that one. Uracil.
Good job. Excellent. Yeah. Because I sometimes don't remember the names, but I remember the letters. You know, A, C, G, T, and U. Okay. So that is correct. So it's just those. Okay. What are amino acids used for?
They're really small organic compounds that are the building blocks for proteins.
That's right. Yeah. Do you remember how many are of how many amino acids are there?
Like 6?
There's a little more than that. Okay. Anna says 22. Okay.
Wasn't it? No, it was 20, wasn't it?
So there's 22 that are the ones that code for in the ribosome to start building the protein. Okay. One of those 22 is only used by fungi. Okay. And another one is used kind of as I believe it's kind of like the end, the end part of the sequence. So it's not used very, very much. Okay. So Spencer says, interesting about lipids because I had severe seizures when I was young and having a fat diet controlled them. Okay. So here we have an example that a diet with fat actually helped the brain function better. Okay, so very good example, Spencer. Thank you for sharing that. Okay, so yeah, so there's about 22 that are the ones that are coded in this process with the ribosome. Okay, there are more than 500 different amino acids that are more rare if you can think of that. Okay. All right. Excellent. So Patrick, yeah. Okay. Okay. So yeah, we need to make sure that your mic is not breathing. Okay, too much. I think now it's a lot better. All right. Ketogenic diet. So it was a diet with 99% fat, a ketogenic diet. That is interesting. Okay. Wow. Why are there only 21 on the amino acid chart? Okay, the reason why in this chart we have only 21 was because of that. The 22nd is, I believe, one that was only using fungi. And so they decided, all right, we are now going to use that, okay, to put that in the chart. All right, that is if I remember correctly. All right, Griffin, you are now open mic, so mute yourself, okay? All right, good deal. So now let's go and do the review if you wish. This is going to be a little different, Maria. So let's see, we go and see this animation, okay? What is the yellow and green? part there in the animation? Can you all see the animation correctly? What is the green and yellow parts there? Oops. Sorry, I switched the window. I lost the panel for the question. Maria, are you still around?
Yeah, I'm just trying to rack my brain for the answer.
Oh, okay, no problem. Okay, here I'm getting this back. Oh my goodness. Yeah. Okay. So Jen says it's the ribosome. Okay. So that's correct. So the red and yellow part is the ribosome. Why is it in two colors? Anyone? The smooth and rough is on the endoplasmic reticulum, okay? So they can distinguish one another. Let me tell you why the ribosome is generally represented as two parts. They basically said that the ribosome is composed of two subunits. Each unit is a protein, okay? So the yellow one is one subunit, and the green one is another subunit, and the ribosome only works when the two subunits are working together, okay? Andrew says that he can't hear me. Is anyone else having problems hearing me? Okay. Okay, so we are okay there. All right. Okay. So Griffin, I already unmuted you. You don't need to be a panelist. Okay. All right. Thank you for that. So the ribosome is made of these two proteins, and that is an interesting thing that is important to understand, is that the proteins, many times, in order to work, they have to join with other proteins. Each one of these pieces, okay, the blue and the dark and the light blue are other proteins that have specific functions, okay?
I hate that.
Okay,
you
Patrick,
know what
I
it's
unmuted
doing,
you.
do you?
So mute yourself.
Okay, tell me what it's doing.
Okay. All right, thank you. All right. You need to make sure that you control your mic on your side, okay? Okay, so what is this, Maria, what is this string that is going through this part, this little black one? What would that be?
Is that RNA?
That's right, yes. So that is the messenger RNA. And why is it going through the yellow part? What is going on there? What is going on with the messenger RNA that they move?
Is it translating to protein?
Yes, that is correct. It's translating to the protein. Okay, so, and it goes, can you see that it goes like 3 little pieces at a time? What are the dark blue pieces here in the animation? the dark blue ones. This one here, okay, that come in, attach here to the messenger RNA, and then they leave. Anaphase tRNA, yes, so it's the transfer RNA, okay? And what is this part here building up? What is this string? Wiggly line, Maria.
That's RNA2 coming from the top, isn't it?
So this one is the sequence of amino acids, okay? So the transfer RNA brings just one amino acid, and it starts building the sequence of amino acids to build a protein, okay? So Jens and Jacob says it is the protein building, all right? Yeah, so the protein is being built there, okay? And it kind of goes out of the ribosome. Today, we are going to talk about what is this part. What is this? These squiggly lines, they look like a membrane, and the gray part, all right? But in the previous lesson, we talked about how the transfer RNA brings here the amino acids, and they start building the sequence of amino acids to build a protein, all right? Now, this animation is extremely slow, okay? Imagine that this, first of all of these things are at the molecular level, all right? So they are very, very small to begin with. But they happen really, really fast. That is something that also blew my mind that I didn't know, really. Okay. So excellent, Maria. Thank you very, very much, all right, for helping us with the review of the lesson. So now we are going to turn the time to Chas to tell us about the academic concept number 1. Okay, and Maria, you are for 2, so you can prepare to come back. Okay, Chas? Mm-hmm.
Can you hear me?
Kind of, yes. Mm-hmm.
Okay.
Mm-hmm.
Do I just read it out loud?
More than reading it, I would prefer if you want to kind of explain what you learn about it. Okay?
Okay.
This concept. Mm-hmm.
The central dogma of molecular biology is DNA makes RNA, and RNA makes protein. It is an amazing process that God has designed to create all the variety of structures and functions for all living organisms. Several organelles in the cell participate in the process of protein synthesis. Proteins make thousands of different structures within the cell and outside of the cell. Enzymes, hormones, and biology machines that perform the cellular functions of living organisms. Just think about it. Each cell nucleus is like a library that contains.
Olson, can you mute yourself? Mm-hmm. Thank you.
The chromosomes made DNA a delete copy of a whole instruction manual for making all the proteins needed, all the biology functions. To allow the organism.
So, Chuck, instead of reading it, I actually want you to tell me what you think about this. Okay.
Okay.
Mm-hmm. Yeah.
Like how the DNA is like built by like how the protein like helps us like keep going and stuff. Like, you guys
Okay,
think
so
that
I have a question. What, I may kind of hear a comparison between DNA, RNA, and proteins. Okay, so what is the DNA? What is the nucleus like?
The nucleus is like the main concept of what?
Like what? Sorry.
I mean I don't know.
Let me try another thing, just okay. So think about this. The nucleus contains the DNA of the organism, all right?
All right.
All the information, all the genetic information of the organism is contained there, okay? Now, the DNA by itself is not useful, all right? The DNA only is useful as the instructions for making something else. Okay. So it's, for example, when you buy, let's suppose that you buy a Lego set. Okay, you go to the store and you buy a set of Legos. What helps you to use that Lego set?
To build like a structure?
Yeah, the instructions. Okay. So are the instructions the Lego set?
Wait, what?
Are the instructions made out of Legos?
No.
No. What are the instructions made out of?
Paper.
Paper, yeah. Printed paper, and words with pictures. They have pictures of Legos, but it's not like that. Okay. All right. Okay. So let's suppose that you have a whole shelf full of instructions, okay, of different Lego sets, how to build this and that and everything, and you put that all together into a library, okay? What would be like that if you were to compare that to the cell?
Like how everything's organized.
Okay. What do you think, when you go into your house, where do you put, let's suppose that you have printed material, okay, like books, where would you put them?
In my shelf.
In where?
On my shelf.
On a shelf? On a shelf, yeah, on a shelf, on the library, okay? So you have a library for all the books and everything. So the cell nucleus is like the library of the cell. The interesting thing is that each cell contains a whole copy of the whole library. All the instructions to make all the different things that you can imagine to make with Legos. Okay. Each little book of instructions that helps you to build one specific artifact of Legos, okay, would be like a gene. Okay, the gene is just a set of instructions that help you build one thing of Legos. Okay. All right. Now, suppose that you cannot take things out of the library. Okay. You cannot check things out of the library. You can use them, but you cannot check things out of the library. Everything has to stay in the library. What would you do? what would you do in order to actually use these instructions?
Copy it.
You copy them, yes. What would be the copying of the instructions in the cell?
It like forms another.
Sorry, what is it?
It forms another.
I couldn't understand. I'm sorry.
It forms another.
It forms another. Okay, yes. And so we said that the library is the nucleus. The DNA is the books in the library. Okay, and a portion of that DNA, the gene, is the instruction for one thing. Okay, how does the cell make a copy of the DNA? You can, the rest of the class, you can help out also by typing what you think the answer would be here in the question panel. Okay. Thank you. You can see the question panel also, Chas? Mm-hmm.
I don't know.
Kind of. So you're doing fine, Chas. Don't worry. Okay. This is okay. So the RNA is actually what makes a copy of the DNA. And in this case, it's specifically called the messenger RNA or mRNA. Okay? So the RNA is a copy of a portion of the DNA. All right? Just the portion to build something. Okay? And then we go and we actually use that portion of RNA, and what does the RNA make? Yeah, James, not DNA. DNA cannot be done the other way, okay? If everyone, Emmeline and Connor say protein. Okay, can you see the concept there, the academic concept? DNA makes RNA, and RNA makes protein. Okay.
Okay.
So that is we said that is the central dogma of molecular biology, and this is so important to understand because think about it. The DNA is really the whole library. It's in the nucleus. The nucleus is the library. So the DNA is all the books to make something. All right?
All right.
The RNA is just copies of the little pieces in the library. And then what you get to make with that is protein. And protein makes everything else. All right?
All right.
Okay. All right. Excellent, Chas. Thank you. I hope that this wasn't too traumatic. Okay. You did good. All right. And I understand this takes time to get used to doing this type of presentation. All right. But you're doing good. Okay. Maria, thank you for volunteering. Maria, are you up to... yeah, like I said, overwhelming. I don't know if it's overwhelming doing the presentation or actually thinking about these things. It's really, really cool. Very, very cool. Thank you, Connor. Okay, so Maria, let's do academic concept number 2.
Okay, yes. Okay, so like we had talked about, amino acids are the building block of organic compounds. And amino acids are composed of amine, I don't know how to say that. And and carboxylic acid. And they're kind of like, it's kind of cool because they're kind of like batteries because they have positive and negative. And um, the key elements of amino acids are carbon, hydrogen, oxygen, and nitrogen. And even though there are a bunch of other elements found inside all of these certain amino acids, those are the ones used the most.
Yes.
And what I think is cool is there's 500 different amino acids, but only 22 amino acids are actually added onto the amino acid during the process of translating protein. And so when an amino acid is added to the chain, on the other side, the amine side, a new one joins the carboxylic side of the previous one. Because it's by losing water molecules, which is H2O. And I thought what was also really cool is that the chain, it has like a backbone, like of protein.
Yeah, this is the backbone here. Okay. Excellent. Keep going.
And I also learned that each amino acid's side chain has a specific geometric shape, and it has specific characteristics, like I didn't know exactly what these were at first, but hydrophilic or hydrophobic is some of the characteristics of amino acids. And when the chain starts to grow, neighbor amino acids start to attract with each other or repel each other like batteries, or magnets. And that was cool.
Yes, exactly like magnets, exactly that way.
Yeah, and they form bonds with each other and fold into three-dimensional structures. And then that's how you get the protein.
Amazing. Okay. I find that is just incredible, really. Very, very good. Very good. All right. So that is really how the proteins are being made, okay, by these chains of amino acids, okay. Did you have a chance to see that little video that I make with the rubber band? Okay.
Yes.
Yeah, the rubber. Okay. Was that helpful?
Very.
Okay. All right. You know, I was playing one day with some rubber bands and everything, and I thought, that is how it works. Okay. Yeah, and it is kind of exactly the same way. All right. The amazing thing is that the way that the protein falls depends really on the biochemical characteristics of that. Okay. So Maria, thank you very, very much. Now let's see Jens. Okay. Can I have Jens come forward? Okay, and keep us moving here about the formation here of the protein. Okay.
All right, can you guys hear me?
Yeah, so academic concept 3, and I can hear you.
All right. So this concept is really just explaining what pretty much you said, is there's different amino acids that form a protein, and the proteins can attract each other depending on the charges, if they're negative or positive. As you go down, there's a picture of like all different kinds of combinations, and so as there's different shapes and sizes of the amino acids, they make different shapes and sizes that have jobs for the proteins. So like depending on the sizes of like the proteins depends on their job. So like anyway, then the proteins can combine with each other and can like come together and make different structures and do different kinds of things that work better together than if they were just like by themselves.
That's right. Yes. In fact, there are some proteins that only actually work. They actually perform the function when they are together with other proteins. So they reach this quaternary structure. Okay. Yeah, Anna says, so the protein shape determines its function. Absolutely. Absolutely. So the shape determines function. That is a very important principle. Okay. So in this picture here that I have of different examples of proteins, what do you think that are, for example, in this one? Okay, what are the different colors?
Oh, so like the different colors, like do you think maybe the colors determine like their physical makeup, like the chemical makeup? Like kind of
So
like
the colors
different
are,
amino acids?
it could be, okay, my understanding is that the colors basically said the things, the chain of amino acid of one color is one subunit, okay? And then when you put them all together, then they complete the protein to make it a functional unit.
All right, that's cool. So
Okay,
like they
yeah.
come together to actually like function correctly. Like they can't function without coming together.
Exactly, exactly. And they only function when they are put together, okay? See this one here, we have the ribosome. Can you see the two subunits of the ribosome?
I see blue, and then there's like a bigger and smaller ribosome.
That's right, and so
Half of the ribosome.
yeah, that's right. So this one is the yellow one in the animation. Okay.
Okay.
And this bigger one is the green one in the animation.
All right.
Does that make sense?
Yeah.
Okay. But each one of those parts, okay, is even made of many other proteins folded together. Because can you see the different colors there?
Yeah.
Mm-hmm.
Yeah, everything is made out of protein. Holy cow.
Everything is made out of proteins. Okay. And the proteins are basically the functional thing that makes the living organisms work. Okay.
It's almost kind of
So
like a life source. Sorry.
Yes.
Almost like a battery.
It is really interesting. So in the next class, we are going to talk about, okay, how then we get energy. Okay, where does the energy actually come from? All right, so that is going to be a very interesting lesson. But these are basically the machines. The proteins are the machines that make things happen. Okay. Aubrey asked a question here. She asked, what if a protein develops into a shape that it wasn't meant to be, okay? And what will happen with that? If the protein develops into, it's a malformed protein, okay? Anna says, it makes me think of origami. This is the most amazing origami that Heavenly Father has created. Yeah. And of course, yeah, proteins are made out of protein. Yeah. What do you think that happens if a protein is shaped incorrectly?
Well, that's kind of like a hard question because like first, I'm not sure if like they like can they be shaped into like an unusable form?
Yes. Yeah. So if it's shaped into an unusable form, the body cannot use it.
Yeah. So
Okay.
like they probably maybe would try to transform it just to into energy, just to like fuel the body and to like keep the body going.
Yes. So we are going to see now in the next few concepts. Okay. What happens with that? Okay. So actually, I think that is in concept number 6. We will answer this question. Okay. Griffin, I don't know about what you mean by the question. You asked the question, will we have a live experiment that we will watch soon? I don't know about that. Okay. Can proteins malfunction? Sometimes proteins can become bad for the body, okay, and that is what viruses make. So a virus, basically, like these ones here you have 2 examples of viruses, is a huge protein that encapsulates strands of RNA. All right, and then when these proteins inject themselves into a cell, into a living cell, they release this RNA inside the cell. The RNA starts replicating itself and basically producing proteins, and the proteins actually make the cell sick, and sometimes they kill the cell. All right. And so that's why viruses are not good guys. Okay. So that is how viruses happen. Okay. All right. Thank you, Jen. Very, very good job. Also.
Thank you.
Okay. Anna, would you like to do concept number 4? Concept number 4. Anna, you're on. You need me to unmute you? Okay, hold on. That's it. Anna, yes. There we go. Anna, now you are on.
Okay, so you can hear me now?
Yes, thank you.
Okay. Alright, so my academic concept is about the endoplasmic reticulum, the Golgi apparatus, and vesicles and how they help protein. So I found it interesting how protein, it can be used inside and outside a cell. And if it's being used inside a cell, I guess these proteins, they're formed inside the ribosomes, and then if they're being used inside a cell, they can be just released into the cytosome. If it's being used outside the cell, it has to, the ribosome, it actually attaches to the endoplasmic reticulum, or the ER, and that's why there's 2 different kinds of ER, the smooth ER or the rough ER. The rough ER is the ER with all the ribosomes attached to them. Let's see.
So what do you think that this is in the animation? Oh, now we start the animation over. Keep going, and then when we get to that, we'll ask again.
Okay. So what I found from this section is that the ER is where the proteins are further processed and where they're folded and formed. Then in the ER, some more amino acids, they can be added to the protein. So just lots of more fully forming the protein. And then when it reaches the end of the ER, it is part of the ER breaks off into a vesicle, which carries it to the Golgi apparatus. And inside the Golgi apparatus, they do some more processing and changing and checking and folding. And the proteins kind of join together with other proteins. Then after all this, they go into another vesicle, and that's taken to the cell membrane, where it's released out of the cell by exocytosis into the bloodstream, or it becomes part of the cell membrane.
That's right. Yeah. Do you remember in the lesson when we were talking about the cell membrane that there was one picture that had a bunch of, they had the cell membrane and there were proteins embedded into the membrane?
Oh, yeah.
Okay. How did those proteins get there?
By the vesicle transferring it to cell membrane?
That's right. Yes. Okay. So if we have here, the ribosome produce the protein into the ER, then the protein travels through the ER doing other folding, joining other proteins, and so on. Then it went through the Golgi apparatus. Here it is being tested and whatnot. And then in here, before it's being released, the protein is embedded into the membrane. And then it's transferred into a vesicle, and the vesicle goes and attaches itself to the cell membrane, and now the protein is part of the cell membrane.
That is really cool, because I had wondered what those things were. I knew they were proteins, but I never quite realized how they got there.
Now you know.
Yeah.
Okay. Isn't that awesome?
Yeah, that's really cool.
Yeah, okay. So here in the animation, what do you think that this part is?
Isn't that the ER, where the ribosome is attaching?
That's right. So that is the ER and the membrane of the ER, and there's one protein there in between the membrane that allows the protein that the ribosome is making to go into the ER. Okay?
Yeah.
It's really, really cool. Okay?
Yeah.
So, and again, all the light blue things are proteins that have different functions. Like there are some proteins that bring the transfer RNA in, others can push it. This protein here kind of stops the process to attach to the ER membrane. This one just connects, and then this one processes the amino acid chain into the ER to form the protein. Okay.
That's really cool how, good.
That is an amazing thing.
Yeah.
Really.
I think it's cool how protein, it helps to create more protein.
Yeah. So this is literally like the Lego movie where everything is made out of Legos.
Yeah.
Okay. You certainly have RNA and lipids and a few other things. Okay. But that is very, very interesting, really. Okay. So, all right, excellent. Anna, do you have any other questions or comments about the ER or the Golgi apparatus or vesicles?
No, not really.
Okay, excellent. Good job.
Thank you.
There. Okay. Emmeline, would you like to do the academic concept 5? Let me make sure that Emmeline is not muted. Okay. Oh, yeah. There we go. Unmute. All right. Now you are on, Emmeline.
Hi. Whoop.
Hi. Okay.
So I guess I can. Let's see here. So the concept for number 5 is lysosomes break down unneeded substances, and they are the recycling centers of the cell. So what basically happens is that when a protein is malformed, which means that it has some sort of defect during some sort of process up above, what happens is that it goes through a vesicle to a lysosome, and it's an organelle that's membrane-bound and has at least 50 different enzymes and is very acidic. So these proteins in these vesicles having defects, or unneeded proteins, all these different things that aren't necessary, debris, lipids, viruses, and they are basically broken down into the very basic building blocks and are released into the cytoplasm. And so I think we talked about this a little bit before in another lesson about how, they reuse it if they have debris.
Mm-hmm.
But I thought, that was interesting how, nothing goes to waste. And, you constantly see that with Heavenly Father. He's always putting something into use. There's nothing that is wasteful or is not needed.
That's right, yes. So imagine that if a protein is malformed and is not working correctly, usually in the Golgi apparatus, that is identified, okay, it's tested and everything, and if it's not working, then the vesicle conducts that malformed protein to a lysosome, and the lysosome has enzymes, which, by the way, are other proteins, that break down the protein into amino acids. Okay, and then the amino acids are released back into the cytosol and reused to make a good protein, something that works. Okay. Isn't that amazing? Okay.
Well, I mean, I
good
can't
job.
speak for the rest of the people here, but I think it is.
Yes, of course. All right. Okay. Let's see. Let me see if I can show. Yeah. So people who don't recycle still recycle. All right. Let me unmute that. But so here we have a video. What do you think, Emily? You have the microphone. What do you think you are seeing here?
Oh gosh, I mean,
here?
I actually told my mom a little bit that I had a very difficult time trying to picture where everything's going. So I mean, I think I actually need help with this.
Okay. So let's see if we can kind of slow things down a little bit. All right. So this video is going to show how white blood cells, okay, receive signals that another cell needs help. Okay. So let's start and look at things from the beginning again on that. So here we have the blood, okay, the bloodstream, the red blood cells, and these, what would this be if this would be a blood vessel? What are these bigger ones? What do you think, Emily? Any guesses? Those are white blood cells. That's right. Okay. All right. So let's continue and see what happens. Now we are seeing here the membrane of cells, all right, and how they send signals from the membrane of one cell to the exterior of the cell. All these things are proteins that are embedded into the cell membrane. These filaments are part of the cytoskeleton of the cell that gives a structure to the cell. And this, again, is part of the cytoskeleton. All these are made out of proteins. Let's see how rapidly the filaments form. And each one of the blue, purple pieces is one protein of actin that makes these filaments. Then there's another enzyme that comes and breaks down the filament. This is another protein called tubulin. Isn't that a cute name? Tubulin. This is a protein that is moving a vesicle through the cell. Did you remember that we saw that vesicles move towards the cell membrane? How do they move? Well, they move by these proteins. They are called kinesin proteins. Now, here we have other hormones that are also proteins that are released from the cell membrane to the outside. Oh, no, this is a ribosome. oh, sorry, let me think. Hold on, let me back up a little bit. This is actually, let me back up a little bit more. Okay. So we have here the kinesin protein moving the vesicle through the cell. That is a mitochondria, and this is the nucleus of the cell. This is the nuclear membrane, and those are strains of messenger RNA coming out of the cell nucleus. Okay? Isn't that amazing? Okay, so here we have the messenger RNA, and then the subunits of the ribosome come and they attach and they start going through and producing a protein. All right? Then we see how the protein folds into other things. The ribosome goes into the ER and extracts the protein inside the ER. These are the vesicles coming out of the ER. Okay, and then here we have the kinesin moving the vesicles around. This is the Golgi apparatus and how the vesicles move, and now we see how the exocytosis, so that is the process of the vesicle joining into the cell membrane and releasing the proteins outside of the cell. Okay. All right. Now these proteins are going to work together to send a signal saying, we have a problem here, okay, and see how the proteins join together to send a signal to the white blood cell saying, we need help here. And then the white blood cell is going to flatten and is going to go through the epithelial cells of the blood vessels and is going to go to the cell that needs help in order to do the job, you know, kill the virus or whatever it is that is affecting that cell. All right? What do you think about that? Okay.
That was awesome.
That is really, really awesome. So this is what I want you to do, okay? Emily and everyone else, I want you to watch this video with your family, okay, with your parents, and explain to them what you see in the video. Okay, do you think that you can do that now?
Yes, I think I can.
All right, okay. Yeah, so it is, these are really amazing machines. Did you see the variety of proteins and how they you know, someone says the kinesin looks like legs with no arms, you know? Okay, so it is really amazing. It's just, you know, I don't know, just a little bit. It's really, really mind-blowing. Okay. So we finished the whole time, really. Okay. Patrick, we have a couple of minutes. How does this relate, all the things that we have talked to, how do they relate to the gospel principle here that each person can make an important contribution in life?
Well, I got this whole thing planned out in my mind. I was taking a little more time, but that's all good.
No problem. So, if you want to, yeah, go ahead.
I'm just gonna give you the speed version, if that's okay.
Yes.
Alrighty. So, like, what I was thinking about, the first bullet point is it says identify all the individuals in the story of Moses. Well, I'm not gonna identify all of them because that would take forever. But
yeah,
instead, I'm gonna mention one person that actually really stands out in my mind. Moses' sister, the one who saved him from Pharaoh's wrath when they're killing every priesthood holder. I don't think they talk about her too much, but if it wasn't for her, a very, I guess, small person in his story, none of that would have happened. So it's very important that we recognize that. And I mean, she saved his life, freeing the Jews from Egypt, and then after that, bringing them to Jerusalem, and going to where we are today. So
Yeah.
that's important.
That is important, and every single one has a very important part and piece in this. Okay. Go ahead.
And there's this poem here. I'm not going to read it because it's pretty long. I'm just going to tell you that it's pretty much saying one thing does everything for a different reason. And it's pretty much just summing it up. Everything has a purpose. And one person can pretty much change everything. I mean, there's so many examples in history. I'm having trouble picking one, but there's a lot of different people who can change a lot of different things. Like Joseph Smith, when he translated the Book of Mormon. Look at where we are now. And you don't have to, it says here a question, do you have to be something big to make an important difference? Not really. Like the example I just gave, Moses' sister. I'm guessing that we don't even get her name, so.
Yeah. So one thing that I just want to encourage you, all of you guys, is to remember, and you don't have to wait until you are older or until you go on a mission, okay, or anything like that. You can do important things that make a difference now. Okay. Yeah. I may be one, but one becomes two when you stand with me and I stand with you. Hillary Wicks. Okay. Beautiful song. Okay. So, Patrick, would you like to offer the closing prayer for us?
Sure. All right. Dear Heavenly Father, we are thankful for the chance we've had to come here today and to learn about the composition of cells and what makes us all up. We're thankful for the chance that we've had to learn that everyone has a purpose and that you don't have to be something large to make a difference. We're thankful for the chance that we have to learn about these things across the country and across the whole world. And Heavenly Father, please help us to go throughout our day with happiness in our hearts and help us to take these things to heart. And we ask these things in the name of Christ. Amen.
Amen. Thank you very, very much. And yes, it is indeed that our influence is like ripples in a pool. Okay. Very, very good. Okay, excellent. Thank you very much, and we'll see you next week. Okay. I'm going to